Thank you so much, sir, Dr. Bhaskar Reddy, sir. It's my pleasure and honor for me to associate myself with such a huge group, that too for the conservation of nature. I am really lucky to be with you or to be associated with you. Thank you for inviting me to lecture on this aspect, that is the most burning problem in Neem, that is the dieback disease. uh, though we say that uh, it's not only one problem, but there are many associated problems also. Today I would like to talk about the dieback disease of neem trees. And actually, if you see, we all know we use neem for many management practices. That is mostly the neem leaves and all used, or I mean the neem fruits and the neem cakes, everything is used for the management. But neem itself is now facing a huge problem. It's not that it's a new problem, it was there earlier also, but now it has spread rampantly throughout India. And if you see, it is neem is also known as Indian lilac or margosa, most eco-friendly plant and evergreen tree, and it is a native of the subcontinent. And especially nowadays, a lot of emphasis is there about the threat of this dieback disease, which is spreading at an alarming rate in different parts of India. And there are this neem tree. I am directly going to the topic. Mostly, this neem tree is affected by different organisms, both biotic as well as abiotic. And if you see, there are two bacteria. There are almost around fungi around 31, and mycoplasma-like organisms are also there. Now we call them as phytoplasmas. And then we also have around insects and pests, almost around 114, and mollusks, and even animals like rodents, and are also playing a role for in these biotic stresses. Whereas if you see the abiotic stress, so this neem is affected by both biotic as well as abiotic stresses. And if you see, there are almost around 150 organisms are known to be associated as a biotic organisms. Whereas in the abiotic stresses, we have a decline and fasciation. And decline is nothing but, if you see any stress plus a pathogen associated like that, these crown region of the tree, it will elongate, and it will, that means all the foliage will fall down and it will appear as a giraffe neck. And this may occur due to prolonged drought also. And another abiotic stresses are fasciation, mostly due to, I mean, small to medium sized branches are there. And mostly with the distorted and increased leaf buds will be there. And another important abiotic stress is the rapid wilting of the leaves. And pollution damage is also known to play a major role. Especially, this was reported from our India only, after the methyl isocyanates which were released from the Bhopal during the Bhopal gas tragedy. This was reported by Ram Prasad and Pandey in 1985. So these are, that means even pollutions are also playing a major role. And we all know that we are going to a very climatic change phase. So this climatic change is also affecting the crops, not only the cultivated crops, but the all animals, I mean the environment as such. So entire foliage, or all the trees, or the crop plants, or the pathogens, all are affected because of the climate change. So all put together have played a havoc on these neem trees. And apart from that, we have also observed the prolonged drought and waterlogging conditions, and prolonged dry spell with intermittent rains, then air pollution. And it is also observed that wherever these construction sites and all are there due to urbanization, there also the maximum trees have been affected. So we know that neem is affected both by biotic and abiotic stresses. And as I was telling, these are two different terms. One is decline, the other one is dieback. So mostly decline is the gradual reduction in the growth and vigor of a plant, okay, in stress, due to which it is caused due to a stress. Whereas if you see the dieback, it is the progressive death of the twigs and branches, and mostly it will start from the tips. And the trees and shrubs affected by the decline and the dieback syndrome, they die within a year, and sometimes they also try to survive. I mean, they rejuvenate and try to survive. So this is, I mean, especially this happens in decline, wherever they can come back to their normal condition of the plant. So this is the difference between the decline and the dieback. And if you see, I am dealing only with the pathogens which are associated with the dieback. Because there are almost around 150 pathogens which are associated, but I want to restrict only to the dieback pathogens. So here you can see the different name, that is leaf spot and dieback, which is caused by Colletotrichum capsicum. And this was first reported by Shankaran, Balasundaran and Sharma in 1984 from South India. And leaf spot and blight by Colletotrichum gliosporoides, and this was reported by Tiwari from Dehradun. And pink disease, twig blight and dieback, Cortesium salmonicola. This also was reported from Rajasthan, and here almost 100% incidence was observed, and this was reported from Bikaner, where there was a high rainfall that was more than 2000 mm with prolonged periods of high humidity. So this pink disease or the twig blight and dieback was noticed. And then we also have canker. This is a Dileep Reddy, which was first reported by Sarva et al. In 1986. He is also from IRA. And wilt, Fusarium solani. So if you see this wilt, mostly this occurs. This was first reported from Kerala, and almost 30% mortality was there. And mostly this was observed in 75-day-old seedlings. And Shukla also reported from 1992 from Dehradun in 2-month-old seedlings. And root rot, if you see Ganoderma, actually Ganoderma appears where the entire plant is gets dried up and all. So it is almost whenever there is an injury or anything, there you can see this Ganoderma lucidum. Okay, this was also reported by Tiwari in 1992 from Dehradun. And as other pathogens, the major pathogens, like you also have twig canker and leaf spot, that is Phoma joylana, and this was reported by Singh and Chauhan in 1984 from Punjab city, India. Whereas twig blight and limited dieback, so this is Phomopsis, was reported by Tiwari in 1992, and he reported it as a Phomopsis species only. He didn't identify the species. Okay, so and from Dehradun, and then leaf spot, this is Pseudocercospora subsessilis. This was reported by Shankaran et al. In 1986, and Mehrotra also added it as a leaf spot, which is severe in the month of December to March, when damage coincides with the winter rains. So you can see the different type of climatic pattern where these dieback diseases are occurring. And we also have another important pathogen, that is weeping canker and dieback. This is caused due to Lasiodiplodia. Earlier it was called as Botryoidiplodia. Now we call it as Lasiodiplodia theobrome. And then we also have another pathogen, that is Cladosporium, Cladosporoides. Actually, this was first recorded by Sarabha et al. In 1986 from India. And if you see the major pathogens, we have like Phomopsis and Fusarium and some Colletotrichum also, but also there are some secondary pathogens, especially if you see this Cladosporium. This is a secondary invader, and even your Colletotrichum also is a secondary pathogen invader. So these are the pathogens which are associated with the dieback disease of neem. This is particular to neem. And if you see, now we know that almost in India, major diseases were reported from India on neem way back in 1986 and all, from Kerala, and in 90s from Dehradun, that is UP. and other parts of even from Rajasthan. So thereafter, Mr. Satish, Dr. Satish and his co-workers, they have worked on dieback of neem, which was caused by Phomopsis aerugite, azideracte. And they have identified this pathogen as the earlier, though Dr. Tiwari has supported it, identified it as Phomopsis species. Now they have identified the species which is infecting the neem. And the occurrence of the dieback was first reported from forests of Dehradun in North India in 1992. And later Satish identified and reported the pathogen causing the neem as Phomopsis azadiracte. And mostly the disease symptoms include twig blight, inflorescence blight, and fruit rot. And later, Dr. Nagendra Prasad et al. In 2010, they have also characterized molecularly by using both conventional and molecular methods. And this is the pure culture of your Phomopsis. And they have collected the isolates from the Bangalore region, from Karnataka region, and then they have identified the genus-specific trimers which were used for the detection of this Phomopsis azadiracte, which is a causative agent. And here it was almost 141 base pairs of DNA was isolated from the diseased species, and that is how they have identified. And this is the PCR DNA, which where you can see this is the base pairs of these different isolates. And here, Phomopsis azadiracte contains two types of conidia. One is the major conidia and macroconidia and micro. And here, two types of spores will be observed. And here, this pathogen is very highly variable. And because of these environment and ecological conditions, it also varies based upon the geographical regions. So that is the reason they have collected the different isolates from different regions, and then they have molecularly characterized this pathogen. And mostly the understanding of this pathogen will also help with spreading of the disease, and thereby by protecting the healthy neem trees. And later, mostly because they are widely distributed, neem trees are widely distributed all over India. So the geographic information and global positioning system, this can be used for the management of the tree diseases. And a survey was conducted by the team of scientists, both in Karnataka, Tamil Nadu, using the global positioning system, this Garmin 12, and they have monitored both the diseased as well as the healthy trees. And here, mostly the details of individual trees such as height, girth, age, latitude, and longitude of the diseased areas which are surveyed was taken, plotted on a map, and then map info was the software they have used. And the conclusion was that the severity of the dieback disease was independent of the tree despite of its age and size. Okay, so that means it is known to occur at all stages of the tree. And very high incidence of dieback was observed in most places of Karnataka and Tamil Nadu, and almost 100% in most places, irrespective of the climatic conditions. So this was their observation, and we can see these are the symptoms of dieback tree. And majorly, the symptoms are more, as I told just now, irrespective of the age, size, and height, the disease is known to occur, and most pronounced during August to December, and observed throughout the year. Mostly these symptoms are known to start with the onset of the rainy season, and they are known to later they become severe, and they also continue up to the winter season. Mostly initially the terminal branches are affected, and the disease results in progressive death of the tree. We can see many trees have almost succumbed to death also. And twig blight is a major symptom, and the inflorescence blight, fruit rot, and inflorescence rot, and even 100% fruit yield loss was also affected. And why such concern is that it is also affecting the fruits. So most of us, the local, I mean, especially for these fruits are used for preparing the neem oil and the neem cake and all. Now we will be suffering a lot, and 100% yield loss is expected from such neem trees. So that is a major concern also, and not only that, we know that neem also has many medicinal properties. And the disease mostly spreads through the conidia, which I have shown just now. The two types of conidia are there, and that because of this rain and all, they are disseminated very fast through raindrops, and the pathogen is also seed borne. So here it is not only seed borne; it also spreads through the seed from one area to the other. So that is the major disease symptoms which we can observe on this dieback. And you can see on the young trees, these are the type of symptoms we have observed. You can see the tips are affected. And here, these dried trees opposite to the healthy brown twigs will be there. And these are some obtrusions which we observed on the affected areas in the young trees. And we have also gone through our veterinary. We have also some Neem Garden over here in our veterinary college. Myself and Dr. Krishna from Agro-Forestry division, we have gone there, we have seen, but it was very much devastated, because one thing which I observed was these are closely planted trees. And when I have gone through the literature, it was also observed whenever there is a wind-blown rain and all, the branches are known. Thank you, sir, for giving me this opportunity. I agree with Dr. Narsimha Reddy, sir. Definitely, we have to first survey the areas, what are all the, how much of the disease incidence is there. Second is, we are planning to, I mean, we are in the research and teaching, so we are planning to give such type of work to our students also, so that there should be an initiation. And one more proposal I would like to have is, now the drones are there. Maybe we can make use of the this software, I mean, precision technology for assessing the disease, and also for even for taking up imaging techniques and all for such type of, because this is a very important crop. So this is my only concern I wanted to put forth before you, sir. Thank you.